So different species might do things differently, but more likely there's evidence even in the same cell that both things are going on. So if we label we do a pulse chase experiment, so you know how to do that experiment. So all the ER proteins are labeled at the same time. And then you measure what happens at the plasma membrane. You see that proteins arrive at different rates. So it looks like some proteins are going slowly, stack by stack, and being modified. But there might be also a more efficient track that other proteins take, and there may be vesicles also going in the anterograde direction. We don't know what those coats are or much about them, so there's still things to be discovered and worked out in cell biology. Maybe one of you will elucidate how that happens. Questions? Okay, so let me show you a movie that sort of visualizes this maturation model and what's going on. I usually take out the sound, but I think in this case it's useful. So this is a movie explaining how proteins go through the Golgi apparatus and this maturation model. We have this movie as well as other movies on Canvas in the different modules that you can look at to sort of visualize what we're describing in class.
The Golgi apparatus consists of general components. The cis cisterna nearest the endoplasmic reticulum, or ER, the medial and trans cisternae, and the trans-Golgi network. Other key players in this process: translated proteins are encapsulated in vesicles in the ER.
Two-coated vesicles.
A group of these vesicles fuse, and these fused vesicles form the cis cisterna. As the protein moves through the stack, it is modified by resident Golgi enzymes at specific locations in the apparatus. These modifications are important because they provide the signal that determines the final destination of the protein. So how does the protein move through the Golgi? Movement occurs in waves. First, the cis cisterna becomes part of the medial Golgi cisternae. Behind it, a new cis cisterna is formed by the fusion of vesicles from the ER. Meanwhile, one of the medial cisternae migrates and becomes the new trans cisterna. Collectively, this process is known as the cis maturation model. Proteins are sorted within the trans-Golgi network. Proteins with the same target sequence are destined for delivery to the same location. The trans-Golgi network then buds off into vesicles. These vesicles then migrate to their target location. These locations include internal organelles, such as the lysosome, the digestive organelle of the cell. The vesicles can also be targeted to the cell membrane, where the targeted protein can be released from the cell for delivery elsewhere in the organism.
Okay, so it's hard to describe in words the maturation model, but I think this movie really helps you visualize how everything is going, why we don't end up with billions of stacks in there, and also where this model totally just is very, it didn't show you the retrograde transport back of resident enzymes, which is essential for the maturation model. And it also was pure maturation model, but it helps you visualize the proteins going through, and also brings up our next topic, which is what happens in the trans-Golgi network. So before we get to the trans-Golgi network, are there any questions? Yeah.
We talked about how vesicles don't require energy input, right? So it's energetically efficient then that the vesicles, the enzymes would move back?
Okay, great question. What the energy that you're referring to is the energy of vesicles fusing with the bilayer, right? And that fusion with the bilayer happens because of the energetically favorable binding of a V snare with a T snare that will then fuse together and allow that, and bring those two bilayers really close to each other so that they can fuse. That's the energy. I don't want you to confuse the GTP that we talked about being required in the initial coding right there, because that's GTP, so it's not energy. I want to make sure you're talking about the fusion, which you were, but I wanted to clarify that for everybody. And now I've forgotten your question because I've talked too long
It's
about it.
energetically efficient for the Golgi to mature the way that it does, and then the vesicles.
Okay, cool. I've never thought about the energy there, and I don't know the process that drives all of that, how a stack goes from one to the other. Do you, Arturo? I think there's some kind of matrix, but I don't know, any kind of movement would presumably require energy.
Yeah.
Maybe there would be a motor, but I've never heard anybody describe that. Maybe that's another thing to be discovered in cell biology. Go ahead.
Well, just to throw more unknowns there. So the movie showed that the vesicles are fusing with each other to build one cis Golgi,
Yeah.
and then that cis Golgi becomes a medial Golgi, which means at some point they stop fusing with that forming structure.
Yeah.
So they say, okay, enough. Now you move up, and now we're going to start building another one. So why does that happen? How does regulated?
Yeah. Yeah, there's a lot to figure out. You know, in biology we present, well, maybe in all your science classes, sort of the end story, but there are so many unknowns still. Which makes science fun. Other questions? Okay, so let's talk then about this, I think what we're going to cover now. Yeah, is this next step that they referred to in the movie, which is we know how proteins get through the Golgi. We know what happens to them in the Golgi. How do they then leave the Golgi? And so what we're going to talk about is the trans-Golgi network, which is a sorting station where there are vesicles that bud from the trans-Golgi network, but here we have many different kinds of vesicles that we're going to talk about. So here's a picture. We have, so we've covered from the ER up to the cis-Golgi network. This is a very old slide, so these question marks with COP1 coats right here are, they have COP1 coats going this way. Nobody believes that anymore, so we don't believe that there are COP1 coats going that way. All the evidence points to COP1 coats going in a retrograde direction, from the Golgi back. So cross out those COP1s right there, because that's known right now. But I like this slide because it sort of puts everything together and then shows you at the trans-Golgi network where different vesicles, how it's a sorting station. And we'll look at how some vesicles will end up going to lysosomes and other vesicles will end up being secreted. So let's look further at this trans-Golgi network. And this is from another book as well, but again shows you the possible paths that happen at the trans-Golgi network. So we're focusing at the trans-Golgi network right here. And there are 3 basic paths. So let's look first at a protein, that white circle up there, that's going to be, right, see that little white circle right there? That's a secretory protein that's going to be secreted to the outside. You all know that that protein had to begin with an ERSS, right? And it began like in the ER. It got modifications there. It got modifications as it moved up through the Golgi. And then in the trans-Golgi network, so it was always in a lumen. It was in a lumen of the COP2. It was in the lumen of the Golgi. If you picture the vesicles budding and fusing, it's never experiencing the cytosol. And then when a vesicle buds right here from the trans-Golgi network into a vesicle, that secretory vesicle will then, when it fuses right there with the plasma membrane, so the final vesicle, that bilayer will become continuous with the plasma membrane. It will fuse there because the right V snare will recognize the right T snare on the plasma membrane. And when that fusion happens, you can totally picture how that will be released into the outside world. Extensively modified with carbohydrates, et cetera, protecting it from the outside world. Similarly, in green right here, that shows a protein that is embedded in the vesicle. It's embedded in the vesicle bilayer because when it was synthesized in the ER, it had some kind of start transfer or stop transfer sequences. This protein could span the membrane a single time, could span the membrane multiple times. And then this, the portion that was facing the lumen of the ER also received lots of sugar modifications that were further modified when they went through the Golgi. And then when that vesicle fuses with the plasma membrane, that protein will now be on the plasma membrane. So that could be a receptor like the LDL receptor. It could be a 7-transmembrane receptor like the G protein coupled receptors. And what is facing the outside world there will be the part that has all the modifications, because that portion of the protein that's facing the lumen of the vesicle was always in the lumen. It was in the ER lumen, it was in the Golgi lumen, and you have to sort of picture that as that's fusing, what the topology of that protein is. Does that make sense? So the cytosolic portion, the portion that's facing the inside of that transmembrane protein, will not have carbohydrate modifications. It's only what's facing the outside. Okay, so we have secretory proteins that bud from the trans-Golgi network and deliver them. We have another specialized kind of transport that, that is only in some kind of cells. This is a regulated secretory pathway. So this means that proteins that are, so the regular secretory one that I just discussed is constitutive, meaning that it just goes on always. It's always going on. The regulated secretory pathway, these vesicles are held until some kind of signal is received from the cell in specialized cells, and then those will fuse with the plasma membrane and deliver their contents. So one example of a regulated secretory pathway that you all are very familiar with is our neurons. So the delivery of neurotransmitters is basically a specialized regulated secretory pathway, and those neurotransmitter containing vesicles will not fuse with the plasma membrane until an action potential is released. Another example are pancreatic acinar cells that have digestive enzymes, and they will only fuse with the plasma membrane when with their digestive enzymes when the right hormone is released. So they're just two examples of regulated secretory proteins pathways. Those are specialized pathways that don't occur in all cells. It's just some specialized cells have them. All cells have the regular constitutive secretory pathway. So every cell is going to have this because all cells have the plasma membrane and stuff and outside world. And then finally, the third kind of vesicle that we're going to talk about that comes from the trans-Golgi network actually brings enzymes to lysosomes where the lysosomal enzymes reside. So this is part of the secretory pathway as well, even though it's a little bit of a misnomer because lysosomal enzymes aren't secreted, so they're going to live in lysosomes. But those enzymes began their life in the ER, went through the received carbohydrate modification. Same story, they're just diverted right here. So you can see how the trans-Golgi network is a sorting station with all of these proteins coexisting in the trans-Golgi network, and then they're sorted to their different compartments. And that's what we're going to talk about now, is how these proteins are sorted to their various compartments. So questions? Okay. Okay, ready? For phase 2. Mind's refreshed. Stretch. Okay, so we talked about how the trans-Golgi network is a sorting station. We're now going to talk a little bit about the secretory pathway, and then we're going to spend a lot of time talking about the how lysosomal enzymes get to their final destination, and then we're going to talk about the endocytic pathway. So this is